Good afternoon, everyone. Dr. Meenal Bhujangra Wagmar is here in the series of Disease of Field and Horticulture Crops and their Management. Today, we are going to see horticulture crops, and our first crop is mango. More number of diseases are present under the mango, so we will divide the mango disease in two parts. Today, we are going to see its part 1. So, coming to the first disease of mango dieback. Causal organism is a Botrytis theobromii. In this disease, mainly we can see that drying of the twigs and branches takes place. Okay, drying of the twigs and branches takes place. The drying takes place from the tip portion to the base portion, and due to this, drying of twigs and branches takes place. Due to this, proper nutrition is not supplied to the leaves, and due to this, the leaves get turns brown and curl and drop down. In severe cases, we can see some gummosis also takes place in a stem portion. That oozing of gum is come from the bark of the affected branches or trunk. And internal tissue of the branches become a brownish in color. Coming to the disease cycle of the mango, we can see that spores, that is conidia, are released from the pycnidia, and that only goes for the overwintering site. The spores get sprayed by the wind, splashing rain and mechanical movement. Coming to the management, in all horticulture crops, most of the horticulture crops, especially in mango, we will do pruning. What is pruning? We will remove the diseased twig or branch by cutting it below to the infection site up to 7 to 10 centimeter. And after cutting of that branch, we will apply the copper based fungicide paste. Coming to the chemical spraying, we can regularly use the copper oxychloride spray and other systemic fungicides such as carbon dioxide 0.1% to manage the disease. But if you will do this chemical spraying after pruning, it will be more effective. Bodex mixture can be also applied. Balanced nutrition with the help of balanced fertilizers program can be ensured to manage this disease. Coming to the next disease, that is anthracnose of the mango. In this, a sexual stage of the pathogen is a Coledotrichum leosporidius and a sexual stage is a Glomerella cingulata. We can see that on the leaves, small angular color dark brown spots are appear, and in extensive case, these spots get dry, and due to this, due to lack of nutrition, that plant leaf get defoliation will takes place. And we also see such a dark brown sunken spots and water circulation on the fruit also. And in significant severe cases, this lead to the rotting of the fruit rot. So fruit rot is also appear at the severe case stages. Such spots are not only restricted of the leaf and fruit, it also comes on the twig and shoots also, and give the drying appearance and dieback appearance. We can see in severe stages such a angular dark brown water sunken spots are appear on the fruit, and ultimately rotting of the fruit we can see here takes place. Coming to the life cycle, first we will see the sexual life cycle. In anthracnose, mycelium, when your mycelium get terminated, we can see the perithecium is a fruiting structure is formed under this, and this perithecium is released to the sexual spore, that is ascospores, and so sexual life cycle is continued by the ascospores. And coming to the sexual life cycle, we can see that acervulus is a fruiting structure which releases to the conidia, and this conidia will help to carry the secondary infection of the pathogen and lead to the disease. Coming to the management, again copper oxychloride 0.3% and systemic fungicide, that is carbendazole captain, can be sprayed. Bodex mixture also applied at 15 days of interval. But if you do this all chemical management after pruning, it will be more effective. So this pathogen can be travel in up to post-harvest condition also. So before storage, dip the fruit in carbendazole solution 0.05% for 5 minutes will help to manage the storehouse diseases also which caused by Coledotrichum. Coming to the next disease, that is mango malformation disease. Causal organism is a Fusarium moniliforme subglutinis. In this, symptoms are appear in the three phases, that is bunchy top phase, floral malformation phase, and vegetative malformation phase. In bunchy top phase, what will happen? The shoots will grow very short, and in the short shoot we can find small rudimentary leaves are appear. Due to this, that crop get the bunchy top appearance. Coming to the next phase, that is vegetative malformation. In this, from in limited seedling growth stage, excessive branches, vegetative branches are appear. Due to this, grassy appearance comes to the plant, and it shows the vegetative malformation of the plant. Coming to the next phase, that is floral malformation, we can see here that in floral malformation, which majorly we can see under the mango malformation, drying of the head with a black mass is persist for a long time, that lead to the floral malformation. In this, coming to the disease cycle, mainly two spores are formed, that is chlamydospores and conidia. Chlamydospores act as a resting spores, which under favorable condition it lead to the disease cycle, and when host is available. Secondary infection spread by the conidia from leaves and fruit to the other healthy part of the plant. Coming to the management, we can follow some cultural practice such as diseased plant should be destroyed, that is nothing but the field sanitation, and remove the plant debris. We also go to use a disease free planting material. There are different resistant varieties are present for the mango malformation. Very importantly, that its incidence get reduced by spraying of the 100 to 200 ppm NAA during October. And again, one more important practice, that is pruning, that is removing the infected branches or twigs by cutting that branches below the infection site for 7 to 10 centimeter. Here I have mentioned 15 to 20 centimeter also fine. Coming to the spraying, foliar spray application can be done with the help of carpenter enzyme 0.1% and capital for 0.2%. We also can use copper oxychloride for 0.3%. Coming to the bacterial blight of the mango, that also known as a bacterial leaf spot. This disease is caused by bacteria, that is Pseudomonas mangiferi pathover indicae. In this we can find that dark and water soaked angular spots with yellow margin or yellow halos appear on the leaves, and due to this ultimately this leaves is going to dry and defoliation of the leaves is takes place. In severe cases from such spots which turns into black, oozing of the gum is also takes place. Such spots also appear on the fruit part. We can see that the spots are also appear on the fruit part, lead to the cracking of the fruit. Coming to the management, we can go for using the resistant varieties and establish the windbreaks. Windbreaks is nothing but the wherever our mango cultivation is present, around their area we will sow some heighted crop to avoid the mango crop from the suddenly heavy wind, to protect it from the heavy wind. To we use the windbreaks. Again, sterilize the pruning tools with the bleach and prune out infected branches. That is nothing but we have to do pruning with the disinfected tools. Coming to the copper fungicide spraying, copper oxychloride, that is 0.3%, copper hydroxide every 2 to 4 weeks from the panicle emergence to harvest we can spray. Definitely this is a bacterial disease, we should use some antibiotics. In this, in case of active infection, streptocycline 0.01% and agromycin 0.01% at 10 days of interval often recommended. Coming to the biological control, Bacillus subtilis or Bacillus amylocophacins can suppress the bacterial growth. Coming to the next disease of the mango, that is powdery mildew, causal organism Odium mangiferi. In this we can see whitish color mycelium powdery growth occur on every part such as leaves, floral, panicle, fruits, and this all lead to the shedding of the flowers, fruit, and it misshapen the size of fruits. Ultimately, these symptoms also carry some gray color spots or lesions and lead to the curling of the leaves. And on fruits also this powdery mildew growth is occur, which lead to the deformation of the fruit and corky tissue occur inside the fruit. Eventually, all infected plant get dry, and in case of leaf, defoliation takes place, and in case of fruit also, premature dropping also takes place. In this, this is scanning mainly two kind of spores, that is asexual spore and sexual spore. In this, Chasmothecium is present as a fruiting structure, which is almost similar to the Clistothecium, but quite different. This Chasmothecium, after maturity, it can break down and lead to the release of the spores from ascus, that is ascospores. This continue the sexual life cycle of this powdery mildew of the mango. Coming to the asexual life cycle, conidiophore, when mycelium get germinated, conidiophore are produced, and from conidiophore, conidia are released, and that conidia lead to the spraying of the lead to the secondary infection of the powdery mildew of the mango, that is Odium mangiferi. Coming to the management, definitely it is a powdery mildew. Sulfur is one of the best management for that. So fine sulfur at the rate of 0.5 kg per tree we can dust. Coming to the application, that wettable sulfur application, first when infections at flowering stage, we will provide our first dose, and after 15 days we can go for the second dose of the wettable sulfur. Similar carbendazime and carthen and tridiomorph foliar applications also recommended. Coming to the next disease and last disease of this part 1, that is mango spongy tissue. This disease is not caused by any causal organism, but it is caused by some environmental, nutritional and cultural practices, that is high temperature, excessive high temperature and excessive sunlight, and such heat stress during the fruit development lead to the spongy tissue. Again, deficiency of the calcium, potassium and imbalanced fertilization lead to the spongy tissue of the mango. Coming to the cultural practice, early harvesting, that without a proper maturity, if you harvest the fruit, that such poor practice also lead to the spongy tissue. And Alfonso is a mango variety, is genetically more susceptible to this mango spongy tissue. So if you want to avoid this disease, so we have to avoid the Alfonso variety also. Coming to the symptoms, we can see that such a loose and softy watery texture of the tissue takes place. We many times when we bring mango from market also, we can see such a disease symptoms. With that also, pulp become a yellowish and corky, mealy and unpleasant taste is developed from this mango. If you cut the mango, we can see such a corky part in the pulp, which is excess yellowish and mealy. And this generally occur near to the seed of the mango, and that's why it make unpleasant in the taste and not preferred by the persons to eat. That's all about the mango spongy tissue symptoms. So coming to the management, here alertness also play the more uh, important role, that mulching to the protect the root from and soil from the heat, because one of the excess heat stress is one of the factor which lead to the spongy tissue. So mulching can protect the root and soil from the heat, and that also we have that also protect the mango crop from the spongy tissue. And we have already seen premature harvesting is lead to the spongy tissue. So harvest at the optimal maturity, not too early, is one of the good management. We have already seen that in both management we have seen that some potassium, calcium deficiency lead to the spongy tissue management. So proper balanced fertilization of the calcium, magnesium and boron and sodium chloride, calcium chloride, this reduce the chances of this spongy tissue. That is also potassium nitrate can use as a foliar spray which strengthen the cell wall, because deficiency of the potassium, calcium, magnesium lead to the spongy tissue. Other management is a plant growth regulator such as gibberellic acid can regulate the seed metabolism but check the local regulation. So this is all about the management practices can be followed during the management of the spongy tissue. So if you like this video, please subscribe my channel, like my videos and share the videos. Thank you.